Hello, my name is Stephen Griffin, and I'm a virologist, and my lab is interested in the way in which viruses cause cancer. And today I'm going to tell you about some work we've been doing looking at virus-driven intrahepatic cholangiocarcinoma, specifically the way that hepatitis C virus is involved with these processes, with these carcinogenic processes in intrahepatic cholangiocarcinoma. I'm really sorry I can't be there in person. And actually all of the work that's been done really has been mainly done by this fantastic PhD student, Alexandria Kilvington, who's been supported by the AMMF during her PhD. So hepatitis C is a major cause of primary liver cancer. Along with hepatitis B, it's responsible for something like 70% of primary liver cancers. So that includes HCC, hepatocellular carcinoma, and intrahepatic cholangiocarcinomas. And this is just a little cartoon showing you what we mean by that here. And we've known for some time that this virus is able to cause cholangiocarcinoma. And for a very, very long, many years, it was thought that the predominant mechanism involved an indirect causative effect through the promotion of chronic hepatitis and inflammation and resultant cell turnover and renewal. However, it's become very, very clear that hepatitis C also has direct cancer-causing effects by affecting the cancer hallmarks and in creating a pro-oncogenic environment. And this is very unusual because hepatitis C is actually an RNA virus, which means it continues its life cycle in the cytoplasm of cells. It doesn't integrate into our DNA as some viruses can. It doesn't persist in the nucleus or indeed generate a very obvious proto-oncogene. So the way in which this virus does this is really, really important to understand. And what's become even more important to understand in the new era of curative therapies for hepatitis C is actually how the risk of cancer is persisting after patients have been cured from their hepatitis C with these new drugs. So we know that established risk factors such as hepatitis C and hepatitis B infection are well-known documented causes of HCC and intrahepatic cholangiocarcinomas. But it's becoming very clear that patients who have been cured of their hepatitis C and are told by their doctors, you are free of your virus, still actually maintain a relatively high risk of developing primary liver cancer. And this, of course, is a major clinical problem because these patients are being told they're cured, but they're still having to come in for follow-up visits, fibroscans, and what have you, and screening. And the risks of developing these tumors still remains very high indeed. And we're getting close to understanding how this is happening in terms of the mechanisms. Because if you remember, I told you that hepatitis C is an RNA virus and it's not exerting effects directly on our DNA or anything like that. But it does appear as though the virus, through manipulating host cell signaling, is able to leave an epigenetic imprint upon liver cells after it's been cured with these direct-acting antiviral drugs. So this leads us to term the kind of cancer causation that hepatitis C is causing as almost a hit and run type mechanism. It's imprinting its pro-carcinogenic field effects upon the liver, and this then remains once that virus has been suppressed or removed through the action of direct-acting antivirals. So this, for us, was really intriguing because it's a non-invasive way of studying these different types of cancers in terms of we're not having to manipulate the genetic contents of cells. And so we rationalized that if we could get the right systems available to study these tumors with the virus infection, then we could look at this in terms of a non-invasive mechanism and identify causative factors that would equally apply both to hepatitis C-driven cholangiocarcinoma as well as other forms of the disease as well. So it's important to consider the environment in which these tumors are happening. And as I mentioned before, there is this indirect field effect of inflammatory status in a chronically inflamed liver that's being caused by these virus infections. And this is a picture of Prometheus, and it's really being shown here to illustrate the fact that our livers are incredibly unique in our bodies in the sense that they are able to regenerate and replace lost and damaged tissue by virtue of the ability of the liver cells to undergo self-renewal and cell divisions. Now, this can happen to a large extent through initial acute trauma by a limited set of divisions by hepatocytes. This is the hepatic triad here. However, in chronic liver disease and chronic inflammation, there's a different process that does this. And this is through the activation and proliferation and division and differentiation of so-called hepatic progenitor cells or liver progenitor cells. And these reside near the bile duct in this area called the canal of Herring. And what's really important for these cells in terms of understanding the carcinogenic mechanisms of both of these forms of primary cancer is that they're able to form and replace both hepatic and biliary cells within the liver. So this common origin of these cells is being constantly activated and making new cells as the chronic damage to that liver, which can occur over many, many years, remember, in hepatitis C, is ongoing. So we realized that perhaps this could be a major source of carcinogenic change within the liver if that virus was actually capable of infecting these cells. And this is important because we know that the liver is incredibly plastic in its organization. And as we know, the progenitor cell can form hepatocytes and cholangiocytes. We also know that these progenitor cells are the source of multiple different forms of tumor within the liver, because these can arise both through transdifferentiation and cross-differentiation, and indeed direct development as these as cancer stem cells forming different tumors within the liver. So even if you've got this infection that's cleared by these drugs and they're incredibly efficient with something like a 90% different cure rate, it's completely possible that if it can infect these cells, that it's causing these pro-oncogenic changes, which could give rise to these two key forms of liver tumor. So we were really interested in developing in vitro systems to look at that. What evidence do we have that this might be a realistic possibility? Well, we know that despite not expressing receptors for the virus, biliary epithelium can be found to show that they are containing hepatitis C antigens, so showing here the HCV core protein, and indeed you can detect HCV RNA secreted in the bile. And we also know that hepatic progenitor-like cells can be infected in vitro by the virus. But something that we did, and I presented this last year, was to take primary hepatic progenitor cells, which we cloned out of patient livers, and show that we can use using a cell co-culture method. We were actually successfully able to infect these cells with hepatitis C. So we do at least have precedent in ex vivo systems that primary hepatic progenitor cells, which you can see here, nice and mesenchymal and variable in morphology, are indeed able to be infected by this virus. So we have at least a cause to start looking at this. However, this sort of primary system really isn't particularly robust. It's not particularly easy to do. And of course, the level of infection that we see is quite low. We're talking about handfuls of cells upon co-culture. So what we needed to do was develop an HCV permissive robust cell culture system that we could look at in terms of the way that hepatic progenitor cells differentiate. Now, we're very limited with the tools that we can use for hepatitis C virus because it's absolutely dependent on a cofactor called microRNA 122, which is generally lost from most liver cell lines. However, one of these cell lines, HUH7's human hepatoma 7, is actually very much permissive for this. But we took advantage of the fact that actually within cultures of hepatoma cell lines, we get variations in expression of cancer-causing genes. And in particular, you can select for cancer stem cell genes such as CD24 and show that these cells actually, even just within a cell culture dish, have differential tumor-forming capacity. And indeed, if you inject these sorts of cells into a mouse, the tumors that you see resulting are indeed very highly expressive of these tumor-initiating cell markers. So actually what we did to generate something which was emulating hepatic stem cell, progenitor cell, which was not actually transformed, was to select cells that were expressing very low levels of this CD24 cancer stem cell marker. And when we did this, we could see that these low-level expressing CD24 HUH7 cells were actually very, very poorly tumorigenic when we injected quite a low number of these into SCID mice, compared with very high level expressing cells and the parental cell line. These really were very poorly tumorigenic indeed. And indeed, I'm not going to show this today, but when you select out low level expression for CD24, you also select out low level expression for other proto-oncogenes as well. So taking these cells forward as a really useful cell culture model, because we can robustly infect these with hepatitis C, we used a well-known characterized trick in cell culture to trigger the differentiation of these hepatoma cell lines into hepatic, hepatocellular-like cells, and indeed also cholangiocyte-like cells in cell culture dishes. And that is just to add DMSO at 1.8% volume for volume. And when you do this, what you can see over the course of several days is you start to see the appearance of these cobblestone-like cells, which correspond to hepatocytes, or at least hepatocyte-like cells as far as an H387 can go. And when you do this in cells that are harboring hepatitis C, what you see is that the appearance of these cells is not only delayed, but it's also far fewer in number. So it appeared as though, and quite fitting with something that would be pro-oncogenic in a damaged liver that was repairing itself, this virus is resisting differentiation signals to form mature hepatic cells. And I'm not going to go into huge details because, again, I presented this last year, but you can see that compared to our day 1 cells, our non-infected day 9 cells are expressing plenty of hepatic markers. They've dropped out of cell cycle, whereas the virus-infected cells are not expressing hepatic markers and are remaining in cell cycle. This is just a nice way of visualizing it. We've obviously got quantification of all of these things. Now, to cut a very long story short, and this is an entire PhD and several years of a postdoc, we actually discovered that in the hepatic system, so making hepatocytes from CD24 low cells, the virus was doing this by manipulating this signal pathway known as Hippo. And Hippo is a really important signaling pathway involved in liver maintenance and size. And it relies on these proto-oncogenes, YAP and TAZ, which interact with transcription factors to drive gene expression, resulting in stemness, etc. And the way that this pathway works is that cell-to-cell contact, GPCR, and different signals that we don't quite understand from the cytoskeletal arrangements of cells activate this master kinase, MST1 and 2, and in turn that activates this other kinase, which phosphorylates YAP and TAZ and prevents them coming in to the nucleus and mediating gene expression. And it was really important to show that our virus-infected cells were indeed manipulating this signaling pathway. And what I'm just showing that very quickly here in our control cells at day 9, you can see that this YAP proto-oncogene is very much in the cytosol, whereas in our virus-infected cells, it's staying, despite the presence of DMSO, well within the nucleus. And just to summarize the hepatic side of this, because it is not the cholangiocyte that we want to look at, we found that DMSO is causing this differentiation by, in fact, activating these hippo master kinases and switching off YAP-mediated gene expression. One of the viral proteins was interacting with MST1 and reducing its levels in cells. We think this might be the mechanism by which this is happening. YAP and indeed TAZ expression was increased in hepatitis C containing cells. And these cells are therefore remaining in cell cycle, resisting differentiation and secreting factors that can actually confer these sorts of abilities to other cells that haven't actually got the virus in them. Do excuse the drilling next door. And we found that HCV is actually doing this through a FOXM1 mediated mechanism, which also has links to genetic instability. So that's hepatic differentiation models. What about cholangiocytes? Oh, sorry. And we confirmed this by RNA-seq. And what we could see that comparing infected cells with cured cells is there's a huge degree of overlap, characteristic of this imprinted epigenetic change within the virus-infected cells. And you can see that this is mainly due to hippo signaling, as well as things like EGFR, which is our secreted component, a metabolic signature, and lots of different cancer hallmark genes expression. And this is just a summary of these things here, which I won't go into. But the take-home message for this is that even if you get rid of your virus, you are remaining with a signature that is pro-oncogenic in this system, which is emulating very much what we're seeing in the clinic. So now to cholangiocytes, and we really needed a way to make these cells, these H387 CD24 low cells, behave much more like cholangiocyte cells, in order to study the potential effects on intrahepatic cholangiocarcinoma. Now, Alex, and this slide summarizes many, many months of work in different cell lines, in addition to CD24 lows. But Alex managed to develop a system which, based on DMSO to inhibit, to activate hipposignaling, sodium taurocholate, and TGF-beta, she was able to drive cholangiocyte-like differentiation in these HOH7 CD24 low cells. And we can see that these are morphologically very distinct to our hepatocyte-like cells, and these are just non-infected cells I'm showing you here. They've lost cell cycle, and they're expressing cholangiocyte markers as opposed to hepatic markers. So this process is a really useful way, again, to look at our hepatitis C infected scenario in the path going towards cholangiocytes. And this is just, again, showing the fact that albumin expression in the hepatic cells differentiation is far more pronounced than in the cholangiocyte ones. And we can also identify many, many more cholangiocyte-specific markers that are switched on primarily just in our cholangiocyte differentiation models, as opposed to things like albumin, which is much more hepatic in origin. And so we're quite convinced that these HOH7s really are, the CD24 lows really are retaining many aspects of hepatic progenitor cells within the liver by virtue of the fact that we've selected them for this low level expression of these cancer initiating cell markers. So this is really, really nice to behold. And, you know, credit to Alex, this took an enormous amount of work to get right, because obviously all of these things had to be optimized. And we did, in fact, spend a lot of time trying to reproduce things in the literature that really did not work, unfortunately. So again, we looked at these cells harboring hepatitis C. At this instance, where we're using what's known as a full-length replicon, which just lacks the envelope proteins to make particles, to ensure that all of our cells were containing hepatitis C, so we had a uniform distribution in the population. And again, what you can see is that these cells are adopting a very different morphology upon differentiation compared to the control cells. And again, if you look at different expression markers, you can quite clearly see that these are not terminally differentiating in the way that our control cells are. And again, Alex has looked by RNA-seq to look at the differential gene expression compared to controls. So this is hepatitis C virus infected as opposed to cured cells. And again, we're seeing this really quite significant overlap between the infected and the cured cells. And this retains, again, an oncogenic signature compared with controls. And what we found was, one, there are some similarities to the hepatic process, but this really does seem to be a very much different set of gene expression signatures compared to the hepatic differentiation scenario. And one that I'd just like to draw your attention to is this nuclear receptors shown here in red, including this here, which is estrogen receptor number 1. But you also have things like transcription factors, which are primarily involved with hepatic metabolism, as well as these various different epigenetic modifications, which are mediated via primarily the polygome repressor complexes. So this again fits with this epigenetic field effect that the virus is having. And we're modeling that in our in vitro systems. But just remember estrogen receptor 1 for later on, because I'm not going to talk about these HOH7 CD24 low cells at the moment, because we have some much more exciting data that we've been producing just recently. And that includes inducible pluripotent stem cell models generating cholangiocyte-like cells. And the reason we wanted to do this is not only because our CD24 low cells are indeed transformed, but because we can go right the way through definitive endoderm, foregut progenitors, hepatoblasts, which are the equivalent of these hepatic progenitor cells, through to cholangiocyte precursors and indeed three-dimensional cholangiocyte-like cells. And we reasoned that, based on our ex vivo data, that we should be able to infect cells at this stage and differentiate them through to cholangiocyte cells. So these are these iPSCs, these 82 cells. And what Alex was able to do really successfully was take these cells all the way through to make these three-dimensional cholangiocyte-like structures, which all do indeed have lumen structures in there. And they're expressing all the right things. They're not expressing AFP at the hepatoblast stage, which is important because they're not committing to a hepatic lineage. And they're expressing markers of terminally differentiated cholangiocyte-like cells. And this is just some more staining of these terminally differentiated three-dimensional structures. And again, we can look at various different gene expression signatures that we were looking at in the hepatic system and trace their way through the different cholangiocyte stages. And I'm not going to go into details here, but suffice to say, we're getting a really good impression of what's going on in these control cells. So the key experiment, can we infect these hepatoblast cells with the virus? And we have to use this trick to suppress interferon signaling in these primary cells, because hep C in culture is really quite weedy. And we can again cure these cells or not, and differentiate them through. Now, we kept these to cholangiocyte precursors because it's much easier to play with these things in 2 dimensions in the category 3 laboratory. And this was really interesting again, because we could see that these gray bars here are where the virus is being cured, that when we have the virus present infecting either at day 10 out of 16 or day 11 out of 16, we're showing differences in certain key genes such as interferons. And when we cure these cells of the virus, this causes a rebound. So the virus is conditioning these cells through differentiation. Then when we cure it, it's changing things. But actually, again, the gene expression signatures that we associate with hepatic differentiation really weren't particularly important. Now, we obviously do have other manipulations of cholangiocytes here, but I'm just going to focus on one finding for today, just to take home, because it's something that we keep seeing coming up. And that is, again, estrogen receptor number 1. And the reason I'm going to highlight this is because we don't see estrogen receptor number 1 actually being switched on in our standard differentiation system when it goes through to a cholangiocyte precursor. However, when these cells are infected with hepatitis C, we do see expression of this receptor, this nuclear receptor, which, if you remember, was involved in the cured oncogenic signature. And indeed, if you look in the literature, we know that ICCA cells are notorious for expressing high levels of this receptor in relation to standard biliary cells, which express extremely low levels. And we know that estrogen could potentially be a driver of more malignant forms of cholangiocarcinoma, particularly intrahepatic forms. And indeed, even going back to the 90s, people have started looking at tamoxifen-mediated inhibition of cholangiocarcinoma. So we think that our approach, including these stem cells and the HRH7 cells, are identifying gene signatures and targets that we can actually pharmacologically use to target the growth of these tumors. And so this virus is not only giving us insights into its own virology and its viral oncology, it's also giving us insights into tumor formation per se. So we're pursuing this at the moment. We're doing experiments with tamoxifen. We've got the virus going in and out, and we'll let you know in due course exactly what this shows. So just to summarize, we're very certain that in both in vivo and in our model systems, hep C is a really useful oncogenic driver. It's a non-invasive driver of both HCC and ICCA. When we cure this virus, it leads this pro-oncogenic oncogenic epigenetic footprint, and it doesn't remove the cancer risk. And it's not just about inflammation, but infection of these progenitor cells provides a potential mechanism by which this virus can cause both of these primary liver cancers. We know that we've shown, at least ex vivo, that this virus can do this. And when this virus is in vitro, in differentiation systems, we are delaying and inhibiting the promotion of differentiated, terminally differentiated hepatic and indeed cholangiocyte-like cells. In the cholangiocyte differentiation systems, we're seeing multiple factors involved, but these nuclear receptors, including estrogen receptor, do seem to be really quite significant, particularly in light of our stem cell data. And when we're curing them again, we're seeing this same hallmark of an epigenetic imprint. So we hope that we're including, by looking at both our hepatic system, we're identifying targets that we can use pharmacologically to target these cancers. So I'd just like to thank primarily by thanking Alex and of course AMMF for their generous support of her PhD, and all of these people present in the group and former, and as well as some fantastic collaborators and the MRC for some additional previous funding for this. And I'll leave it there and let Alex take questions.